The life of a cell or the cell cycle is a time period from which a cell is first formed from a dividing parent cell to its own division into two identical cells called daughter cells. The cell cycle is composed of two major processes, interphase and mitosis. First, let's take a look at interphase.
Interphase can be summarized as the prepping time period before a cell divides. There are three subphases that compose interphase. They are G1, S, and G2 phases. During the G1 phase, the cell prepares to duplicate its DNA. DNA is condensed into structures called chromosomes. Each chromosome is composed of one strand of DNA with several proteins attached to it. The S phase, or the synthesis phase, is when the chromosomes are duplicated. Each duplicated chromosome has two sister chromatids, which are copies of the original chromosome. The duplication of chromosomes enables the daughter cells to have the same amount of genetic information as the parent. The last phase of interphase, the G2 phase, the cell prepares to enter the M phase or mitotic phase. It is during mitosis where cell division occurs. Mitosis can be defined as the separation or division of genetic material in the nucleus. Mitosis is important for reproduction, growth and development of an organism, and renewal of tissue. There are 5 major steps that take place in mitosis: prophase, prometaphase, metaphase, anaphase, and telophase, working with cytokinesis. During prophase, the chromosomes become more condensed and organelles called centrosomes begin to move to the opposite ends or poles of the cell. The centrosomes produce mitotic spindle, which is composed of microtubules. As the centrosomes move farther away, the spindle lengthens. The sister chromatids are bound together by the centromere. In prometaphase, the nuclear envelope enclosing the chromosome fragments and the mitotic spindle begins to attach to the chromosomes via their kinetochores. Each chromosome has their own kinetochore, evidently enabling the mitotic spindle to attach to each chromosome and separate the sister chromatids. Microtubules that have attached to the kinetochores are called kinetochore microtubules. The centrosomes have reached the opposite poles by this point.
By metaphase, the chromosomes has a line in the middle of the cell, forming the metaphase plate. Each kinetochore is bound by a kinetochore microtubule coming from the centrosomes. Anaphase is the separation of the sister chromatids. They move towards the opposite poles of the cell. Motor proteins found within the kinetochores assist with the separation of the sister chromatids. The cell begins to elongate as the chromosomes begin to move toward the opposite ends of the cell. During anaphase, scientists consider the cell to have double the amount of chromosomes due to the fact that the sister chromatids have separated yet are still housed in one cell.
During telophase, the nuclei of the two daughter cells begin to form. The chromosomes become less condensed and the spindle breaks down into simpler components. In cytokinesis, the cytoplasm divides. Due to interactions between the proteins myosin and actin, a cleavage furrow is formed around the area of the metaphase plate, making the cell pinch off into two daughter cells. They are identical to the parent cell in that they contain the same amount of chromosomes and the same genetic information.
Meiosis is another type of cell division that reduces the number of chromosomes by half. Meiosis produces 4 genetically varied daughter cells. However, meiosis does undergo the same steps as mitosis. The true significance of meiosis is to produce haploid cells called gametes. Haploid is the terminology used to describe that a cell has half the number of chromosomes. Gametes are reproductive cells. Meiosis is composed of 2 phases, meiosis 1 and meiosis 2. Both undergo the same phases: mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis. In meiosis 1, homologous chromosomes separate, while in meiosis 2, sister chromatids separate, just like in mitosis.
In prophase 1 of meiosis 1, homologous chromosomes are pairs of chromosomes that encode similar genetic information. Each homolog or partner that make up the homologous pair comes from each parent. The non-sister chromatids, ones that do not have the same genetic information, can cross over or exchange segments of DNA with one another. This introduces one aspect of genetic variation, a process not found in mitosis. In metaphase 1, the homologous pairs align at the metaphase plate. It is during metaphase 1 where another genetic variation factor comes into play. The homologous chromosomes basically position themselves on the metaphase plate independently of one another, known as independent assortment.
During anaphase 1, the homologous pairs separate from one another. At the end of telophase 1 and cytokinesis, 2 haploid cells are formed, each having chromosomes with sister chromatids.
Meiosis 2 essentially follows the same steps as mitosis in that the sister chromatids separate. The end result of meiosis 2, however, is the production of 4 haploid and genetically varied cells.